Introduction for him. He is a father figure for all the neurosurgeons in country. I request Dr. Banerjee to kindly start the proceeding for the oration.
Ladies and gentlemen, we assemble here again for the Dr. V.R. Khanolkar Oration. Dr. Khanolkar was a legendary pathologist from Bombay. Later on, he became the Vice Chancellor of the Bombay University, which was very uncommon for people in the field of medicine to become Vice Chancellors. I told you about another one, Lakshman Swami Mudaliar. One of his very major contributions in Indian pathology as well as Indian public health was the connection between smoking and cancer, which had great ramifications for the future in terms of developing strategies for prevention of cancer. He was a Padma Bhushan awardee and he died in October 29, 1978. This was established, the oration was established in 1987 by the National Academy to commemorate the outstanding contributions of Dr. Khanolkar, the first president of the Academy. The oration is to be delivered by Dr. Subrata Sinha, who is the director of the National Brain Research Center, which has been established a few years back in Gurgaon. He is going to speak to us on the glial cell and its various problems which we see with the glial cell and various types of pathologies including malignancies. Now, the glial cell, ladies and gentlemen, is a very naughty cell. It's full of mischief. It does not tend to behave in the same manner in various species. Forget about species, even in the same species, it tends to behave very naughtily and very mischievously. And thereby, strategies to treat particularly malignancies arising from these glioma cells has been one of the greatest challenges, continuing challenges in medicine. Could I call upon Dr. Subrata Sinha to deliver his oration?
Good morning, everybody. And at the outset, let me express my heartfelt thanks at being conferred this oration. It is indeed an honor to be speaking in a session chaired by Professor Banerjee. As you know, that I work on glial tumors, and I have had a very close relationship with the Department of Neurosurgery, AIMS. They are my collaborators, along with, of course, neuropathologist Chitra, Professor Tandon, Professor Banerjee, Professor Mehta. They have all been helping me and encouraging me. Dr. Mahapatra, before he left for Bhubaneswar, has been my long-term collaborator, and still we continue our collaboration with other faculty colleagues from neurosurgery. I would also like to express my thanks to National Academy of Medical Sciences and Professor Bajaj for being one of my, Professor Bajaj especially for being one of my mentors and my teacher. Presenting a case as a third year undergraduate to Professor Bajaj is a life-changing experience, let me assure you. It makes an indelible mark in your mind. So, with this, I'd like to come on to the topic of my work, which is which is the laser concept, which is this topic, gene discovery on glioma with the context of molecular reclassification of tumors. And if it sounds like a little bit of Greek to the students, it's actually pretty simple. But before that, again, I am really honored to receive an oration named oration of named in honor of Professor Khanolkar, who is a legendary cancer biologist. And in this world where the distinctions between the subdisciplines of medicine are going to merge when we move into problem solving, he was the director of the Indian Cancer Research Center. Dr. Banerjee has spoken about the rest, and really he has been instrumental in shaping cancer research in this country, along with leprosy, which is interesting. So I will talk about a concept of what is a molecular staging of glial tumors, which could be taken in the context actually of any tumor. First of all, why do we need to define the stage and grade of a tumor? Why does a clinician need to do that? Because by looking at the stage and the grade of a tumor, you can actually tell or predict what is the likely outcome and behavior of the tumor going to be, and what would be the appropriate therapy and likely response. So what we do, we will box them, and we will then define a treatment strategy in relation to this box. So of course we all know there's a TNM classification of tumors, which is not really applicable to brain tumors, and we look at tumor histopathology, and we have low grade tumors which do much better but not well in absolute terms, and we have this very high grade glioblastoma multiforme, which, as in this very neurosurgery centered session, I will not hesitate to say things haven't really improved very much for a long time. But then there's a problem also, in addition to that not all tumors really behave as you would expect them to do. Two tumors which look identical in size, shape, and to the histopathologist look very different. Why do they do that? So again, maybe we think, let's come to the idea that they look upon, they're really different diseases. They have different molecular pathologies, and therefore they are different in nature. So tumor glioblastoma A may arise from an aberration of a particular pathway, and glioblastoma B may arise from an aberration of another particular pathway. And if you are going to now develop therapies which are really based on aberrant molecular pathways, then you need to know how different is A or B. This problem may not arise so much for cytotoxics, but it definitely is important for other kinds of therapy. So we are possibly moving into the era of combining histopathology with molecular characterization in the study of tumors. Already immunohistochemistry, by looking at proteins, looks at the products, but that molecular biology, histochemistry, these are all kind of ways in which we can determine the exact molecular pathology of a tumor. We've already done it for breast cancer. Actually, breast cancer classification has changed over the years. We know that we use anti-estrogen tamoxifen for estrogen positive tumors. We've already done a molecular classification that a tumor which is ER positive is different from a tumor which is ER negative, even though they may look the same. So it's the same concept I'm trying to carry forward. And same way, Herceptin, when HER2 new receptors are overexpressed, so we have this antibody which would be a kind of a specific indicator. So again, we have molecularly subclassified these breast tumors. So coming to the brain tumors, there are these different grades of glial tumors. I will be actually concentrating mostly initially on the progression of the high grade of the low grade to the high grade tumor, grade 2 low grade diffuse astrocytoma to grade 3 anaplastic astrocytoma. And then looking at this mess, which is the grade 4 glioblastoma multiforme. And I'll try to really give a summary or an outline of our work as coherently in a brief span of time. These are just different histologies, we'll not go into that. One interesting thing which has already changed in glial tumors is the story of oligodendroglioma. Now, these arise from oligodendrocytes and these all recur. Of course, the others also do, some of them. But interesting thing has been the demonstration of a concept of loss of heterozygosity. So it seems that there are two, the 1p short arm of chromosome 1, 19q is long arm of chromosome 19, and you see there are, every one of us has two alleles and you can use markers to differentiate. The fish will tell you better, but I am a molecular biologist, so I look at gels. But Dr. Chitra, who is our collaborator, she has wonderful fish pictures of the same. Fish is fluorescent, this in situ hybridization. So here everyone has two alleles which can be marked out. So this is the allele in the blood, this is the allele in the tumor, and when both are different, as one comes from the mother and the father, and you can distinguish the two alleles, then you will get this concept of loss of heterozygosity, that here in the tumor has lost one allele of short arm of chromosome 1 and one allele of the long arm of chromosome 2. And so such tumors do much better. Initially it was believed with temozolomide, but they generally do better. So this is one way by which tumors can be sort of subclassified, and you can, obviously they have different molecular pathologies, and it seems that microRNA is really involved in this process. Some microRNA mediated, long RNA mediated regulation is involved in this process. So what initially what we wanted to do, we wanted to look at tumor suppressor genes and to see whether we could see differences in tumor suppressor between low grade and high grade tumors. Tumor suppressors are genes, they are essentially genes which regulate growth and regulate cell growth. And you would expect that in a tumor, the expression or the effect of the tumor suppressor would be knocked out. It's like knocking off the brakes of a car. And they also, some of them lead to this phenomenon called genomic instability. The cancer genome mutates and is much more fluid than a normal genome. So this process of genomic instability and loss of tumor suppressors and activation of another class of genes, which is called oncogenes, contribute to this process. And when we have loss of a tumor suppressor, you have increased cell proliferation, reduced cell autonomy, you have dedifferentiation, and many others. And there was, most famous of them is p53. And we were looking at a region of the chromosome on the short arm of chromosome 17 where p53 resides. And what we find that there is a problem with the short arm, but p53 is normal. So looking at that, what we then have identified a gene which is even more distal to the short arm of chromosome 17, which is a possible tumor suppressor which is lost in, maybe in addition to p53, maybe independently, in tumors which are high grade gliomas, but not in tumors which are low grade gliomas. And that's about half-half. So half the glioblastoma multiforme seem to have lost this one allele of this tumor suppressor. Glioblastoma, and here we could demonstrate it also by wonderfully by doing microdissection. This wonderful microdissection was done with by Dr. Chitra's neuropathology's lab. So this is the same tumor with heterogeneity. There is a high grade region and a low grade region. And this high grade region shows loss of heterozygosity of that locus. That means one tumor suppressor allele is knocked out, the low grade region does not. So it provides actually in kind of an in situ molecular evidence of tumor progression. Also, interestingly, this loss of heterozygosity is associated with increased cell proliferation in astrocytic tumors. So those tumors, if you just do a simple proliferation index, those tumors which have lost this tumor suppressor arm are more proliferative. We couldn't go much further than that, but proliferation itself is a kind of a endpoint of a more aggressive tumor. Another thing that we have been looking at is chemotherapy for gliomas, this is of course in cells. This is nothing new that will be talking, nothing different, sort of in vivo that we will be talking about. But it seems cytotoxics, p53 mutations, p53 is a tumor suppressor gene. So people believe in general that p53 mutations are more aggressive. Now the tumors which have p53 mutations, or those cell lines which have p53 mutations, actually are more resistant to a certain class of cytotoxic agents. Why? Because p53 is a tumor repair, is a DNA repair gene. So if the DNA repair gene is lost, the cytotoxics are more active. So these are a few examples to show that you can actually have different grades of tumors responding to different things. What we had done in the process much before the microarrays came, also this is now I'm changing track and going into another area of our research, was to use RAPD, which is random amplification of polymorphic DNA, which is a kind of PCR analysis which gives multiple bands instead of a single band. So this way the probes spread right across, and we microdissected the tumors, compared with leukocyte DNA from the same patient, and assessed the changes. And what we see here is that if you compare blood and tumor, blood and tumor, blood and tumor, you see these extra bands which are there in tumor. Sometimes there is a loss of a band in a tumor. So what do these mean? These means that these are changes which are occurring between the blood and the tumor DNA. So these are regions of DNA which have been altered in tumors. So what we did, we found a region of DNA which was present, which was different in blood and tumor in a reasonable number of tumors. Some of them may be just non-specific changes, and we picked it up and we cloned it. And what we find is that this is a region which was located on chromosome 4, this region. And then we found that this is a gene which is actually homologous to a gene which had been identified as FAT1. FAT is actually means fat. It was discovered initially in flies, in Drosophila. And there in Drosophila, if it is lost, if there's a region called the imaginal disc that starts forming a tumor. So it's a kind of a tumor suppressor. So this is a part of a family of genes which is called the cadherin superfamily, and it is found to be essentially in Drosophila it functions as a tumor suppressor gene. And this is a huge number of cadherins that we find in human beings also. And this is classical cadherin, and you can see fat is a huge molecule. And what it does, it participates in cell-to-cell interaction. And when you look at cancer biology, you know that cell-to-cell, the signaling between two cells next to each other is also very important for telling tissues how to regulate each other. Now, it's important for cell migration, and it results in actually it sort of when you knock down the gene by siRNA technology, it reduces cell proliferation. In Drosophila, it maintains the polarity of the cell. In human beings, it has been found to be both an oncogene as well as a tumor suppressor gene. So there are certain genes which can function as either A or B depending on the other mutations which are present in the tumors.